first speaker today is Gail McConnell, Professor Gail McConnell, and she's from the University of Strathclyde. Her team, Mesolens lab, works on designing, developing, and applications for linear and nonlinear optical instrumentation for biomedical imaging. So we're happy to have you. Welcome.
Thank you very much. I'm absolutely delighted to be here, and it's lovely to see so many friends in 4 dimensions after such a long time. Thank you very much to the organisers for having me. So yeah, I'm going to be talking about the Mesolens today, and I realise that some of you have heard a little about the Mesolens before, but I'm probably not the person that you want to really be hearing from. I've got 3 PhD students who are presenting posters at the poster session, and I strongly encourage you to go and hear what they're doing from them rather than Gail's interpretation of events. So I'll tell you a little about what the Mesolens is, why we've developed it in the first place. It's now about year 15 of the project. And I'll go on to explain some new applications, some new technological developments in the field. So I'll start with some introduction. Now, this is not the first stitched and tiled image that we've seen at this particular conference, and it's certainly not the best or the worst. This is data from Kurt Thorne when he was at UCSF, and it's a section of mouse kidney, one of these proprietary slides from Life Technologies, I think they're currently called. And it's been imaged using the stitching and tiling method that Rupert discussed from tissue agnostics in the industry talk just before coffee. And it's satisfactory as a method, except it introduces some aberrations to the image itself. So you can see these dark bands around each one of these individual image panes after it's been registered computationally. But there is also a level of detail that is difficult to see in this, at this display magnification, which is the adjacent panes are not necessarily registered seamlessly next to each other. So there are discontinuities spatially, and that can make it difficult, for example, in post-processing or analysis to, for example, segment cells or cell nuclei. So it's a bit tricky. Except there are very few imaging technologies that can image across very large spatial scales, and that's kind of where we approached this problem from, was how can we, as optical physicists, develop new instrumentation that can allow imaging of larger fields of view and large volumes of capture. So we started by revisiting what can your ordinary microscope objective do. So you'll maybe recognise some of these as Rayleigh resolution profiles, but they're not in specimen space. These are in image space. And what you'll see is that these Rayleigh resolution profiles look almost identical, irrespective of whether you have a high NA, high magnification lens, or a low NA, low magnification lens. And the reason for this is almost entirely historical. And it's because these objective lenses, and almost all objective lenses, were designed to match the visual acuity of the human eye. So with a 10x magnification eyepiece, and again, remembering that this is image space, not specimen space, the visual acuity of the human eye is about 7 micron. And until the advent of modern photodetectors, CCD cameras, CMOS, and so on, there was no point in designing or having a light microscopy objective lens that would generate or produce more resolution detail than the human eye could see, because the plan was that you looked down your light microscope, drew what you saw. We're not in that space anymore. We now use cameras, photomultipliers, SPAD arrays, whatever your preferred photodetector is, to acquire image data. That's not to say that these lenses are redundant. Absolutely not. These can provide very high spatial resolution detail in the case of, for example, the high mag, high NA lens, where we have sub-micron resolution in 3 dimensions, albeit within, admittedly, sub-microscopic tissue volume. Or at the low mag, low NA lens end of the business, you have slightly poorer spatial resolution, but within a larger volume of capture. So what we figured was that the human eye is becoming increasingly redundant in light microscopy. So we designed a lens called the Mesolens that can accommodate more resolution units than the human eye can see. And this is the Rayleigh resolution profile in image space for our lens. So our lens has a rather unusual combination of low magnification and high numerical aperture. And what this allows is imaging with subcellular resolution in 3 dimensions across a volume that exceeds 100 cubic millimeters in total. So the Mesolens itself, I can tell you a little bit about that. This is a cartoon diagram, probably obviously. On the left-hand side here, we have the light path of a conventional light microscope where the specimen sits here. We have an objective lens, tube lens, and rather ineffective human eye as the photodetector at the top here. Next to it, in the right, is the Mesolens shown to scale. So you can see that it's gigantic. It is roughly the length of a human arm, and depending on how much you lift, it's also about the width of a human arm. Each one of these individual lens elements can be up to 80 millimeters in diameter. And you'll see that there are very many of them, and some rather interesting looking elements such as these off-axis compensator plates and dichroics, which are inside the objective lens, which allows us to perform wide-field epifluorescence imaging. There are no nonlinear elements in the Mesolens. We use simply ray optics and a combination of nous, or knowledge, and Zemax to design the lens prescription. However, once we have the lens prescription, manufacture is not trivial. We don't do this at Strathclyde, and it's managed for us by a third party based just outside London. What we do is we take our Zemax prescription, we go to a factory in Wales with a UV laser pen and batch test each one of the glass types to understand whether it has sufficiently low fluorescence for use in the Mesolens itself. What you see in the photograph here is two pieces of supposedly BK7. You'll see that with UV laser irradiation, one is weakly fluorescent in the orange, one is strongly fluorescent in the green, and we can use neither of these. So we need to batch test each one of these glass bulk blocks as possible candidates for inclusion in the manufactured Mesolens. Once we've tested the lenses, the glass types, we then supply it to our third party who do the grinding, polishing and mounting into the metal cylinder that goes on to form our objective lens. I won't take you through each one of these parameters in full, but effectively what we have is a lens that's been designed to accommodate a 6mm diameter, a 3mm working distance, and with subcellular resolution throughout that entire volume. We have multiple correction collars that allows for imaging of different immersion fluids, oil, water, glycerol, and we can form an image in air. It's just that the resolution profile isn't quite as sweet as we would like it to be. Unlike most other mesoscopic imaging methods, it's also chromatically corrected across the entire visible spectrum, which means it can be easily used with any of the conventional photoproteins, or fluorescent dyes or indicators that the biologist wishes to use. Because we have more resolution units per unit area than most conventional lenses, we end up with a lot of data. So each one, if we assume nyquist sampling, naturally, roughly each two-dimensional image is about half a gigabyte, and then you put in a couple of colours, so you're at 1.5 gigabytes, and then you think about the volumetric imaging. We need maybe 1,000 images in the Z direction, so that takes us easily to terabytes, and that's with the fixed samples. So the amount of energy that's required to manage your data is increasing only exponentially. I mentioned here that the time taken to build an image is entirely variable, and I'll do my best to remember to explain how that manifests as we go through. So when we conceived of the Mesolens, that 6mm field of view and 3mm working distance was not an accident. We'd spoken extensively to the Mouse Atlas community, and we had envisaged that they would be one of the main users of the technology. And a mouse embryo, when it's day 12 and a half, is exactly 6mm long, and it's about 2.5mm thick. So that's how we arrived at the design specification, and then with the resolution we wanted to see inside every cell of that type of specimen. So we began by taking our Mesolens, because we had one at that time, and because it's too big to incorporate in a standard microscope, we built the microscope around the lens itself. So we used the ordinary point scanning confocal principle, where we have a laser illuminator. We have a pair of scanning galvo mirrors, except in our case, the scanning galvo mirrors are roughly the same size as a car wing mirror, because we need to overfill the back aperture of the 60mm entrance to the scan lens and Mesolens themselves. We then detect through the fluorescence or reflection back up through the optical path, and we use a confocal pinhole the same way that your proprietary or commercial confocal microscope does. So this is one of our Mesolenses here. This is the front optics here, with eyepieces obviously here. We have a liquid light guide that pipes in our cool LED illuminator, for wide field epifluorescence. What's not shown here is a camera, which normally sits at this port here, but I will describe that camera in a later slide. So these were some of our first data. What you see here is this mouse embryo specimen that I mentioned previously, and it's dual labelled. So what we have are acridine orange labelled cell nuclei in yellow, and we've got an antibody against a neuronal variant of beta tubulin shown in blue contrast. With each successive image that is played in this movie, we're moving the specimen relative to the front element of the mesolens by 3 micron position in Z. We're sampling over only 200 micron. But what you will see, if we can play the movie again, please, thank you very much, is that new detail is resolved at depth. So for example, these, whoa, I've went back, there we go. So these ribs that are here will disappear, the liver will become more prominent, you'll be able to see the edge of the brain, and this region here being the eye. So we have a now macroscopic view of this multi-millimetre sized specimen. Except this doesn't really show the resolution performance of the mesolens. So any zooming I'm showing you is entirely computational, where we've cropped at a region or we zoom in computationally. The mesolens is not a zoom lens, which means that we do have a fixed relationship between numerical aperture and magnification. They do not vary, unlike in the stereo zooms that you might find in your own labs or core facilities. So what you see here is just I've cropped out the region that surrounds the eye of that previous data set. So that was horribly downsampled for presentation. This is raw data, so there's no post-processing except for a bit of cropping. And what you see here is the edge of the lens of the eye. And if we can play the movie, please. I was expecting Star Trek. Okay, so what we have is the edge of the retina here. And we're imaging only through about 200 micron deep, but that has collapsed, and eventually it will go on to form the optic nerve. We then kind of slightly started moving away from mouse specimens because the Mouse Atlas folks knew about it, and we're still seeing mouse embryos coming through the lab. But we realised that there were other model organisms. So, for example, we developed protocols for fixation, staining and clearing of whole adult Drosophila and tsetse flies, and we're now receiving samples from infection labs worldwide who are interested in using the mesolens to map, for example, the trypanosomes within the intact tsetse. I mentioned previously that we have a camera. Something that we did not see coming was the growing need for wide field of view in 2 dimensions rather than volumetric imaging. But because of the number of resolution units per unit area of the mesolens, we needed a camera with hundreds of megapixels, and, well, frankly, none really exist unless you're an astronomer with very, very deep pockets. So what we eventually found, thanks to our friends at Teledyne, who gave us a call and said this might work for you, even though they weren't selling it, so kudos to them, is a Ccd sensor, 28 megapixels only, but it's located in 2 orthogonal piezoelectric transducers. And when we apply a voltage to these, rather than moving the sensor by, for example, a full sensor width, we move it by a fraction of a pixel only. So we move it through 9 by 9 positions. So we take 9 images, we perform an operation that looks a lot like deconvolution, and then we end up with an image, a resultant image, that has 9 times the number of pixels, and each pixel is 1/9 of the original size. And this works quite well for us. We get about 250 megapixels, and we can reduce our field of view to about 4.5 millimeters or so, and then we get full Nyquist sampling across the field of view. And I will come back to the camera and how we've used it later. First, though, I would really like to spend the rest of the time giving some detail about new developments and applications, and this is both technology developments and applications in biology and medicine. So the first is kind of in 2 parts, and it's the use of the mesolens for studying host pathogen interactions. And I think that these are the only data I'm going to show you that are published. Everything else at the moment is currently unpublished. We were working with colleagues at the National Institute for Biological Standards and Controls who wish to study and map tuberculosis infection in as much intact lung tissue as they possibly could. So what you see in this still at the moment is a whole lobe of mouse lung. So somebody has pressed play, thank you very much. The initial field of view, 6 millimeters in diameter. So computationally, we're now zooming in. Each one of these little orange dots are auramine orange and rhodamine B stained TB bacilli. So we were able to map the whole lobe of mouse lung and screen it for infection. Obviously, we have physiologically relevant data sets that can back up our analysis. But what we did have to do was develop an image analysis pipeline that could manage these prodigiously large data sets. We were therefore able to then go on and perform principal component analysis and cluster analysis to understand the heterogeneous spread or otherwise of that infection. A very different host pathogen study is with colleagues at the University of Glasgow. So at this level of display zoom, the cyan blob that you see, the cyan contrast comes from DAPI-stained cell nuclei, and we use this simply for landmarking. So the field of view again is 6 millimeters. The working distance here, or the image volume, is about 2.5 millimeters. And if we can press play, please. We're going to zoom in through some rendering artifacts, and you're going to see some red blobs. So these red blobs are trypanosome parasites, so it's T. brucei, and they constitutively express the photoprotein TD tomato. So the researchers that we're working with at the University of Glasgow, similar to the TB in some regards, want to know exactly where the infection is. But the hypothesis that they're testing is, do these parasites cross the blood-brain barrier through the brainstem? So we naturally look at the brainstem and see if there's any clustering of the parasites within that unusually large tissue volume. And what we see is there's not. So that is leading us, at least at the moment, to suggest that it might not be that mechanism that's helping these parasites to invade. Can we press play again, please? Yes, okay, thank you. However, it turned out that there was a side effect, and it was a very good one. And it's the high resolution that the Mesolens gives us. So you'll notice that not all of these parasites are the same shape, nor are they the same size. And it turns out that because of the subcellular resolution that the Mesolens affords us, we can now not just look at where the parasites are, but using their morphology, we can identify exactly where these parasites are in their life cycle, and therefore get a better handle on exactly how the infection has manifest. We're looking at a time point here, day 21 after infection. We can now look at other time points and understand in these clarified mouse brain specimens exactly how the disease has spread. A very different application is clinical, and it's one of the first clinical applications of the Mesolens in our hands. The hypothesis that we are testing here is relating to the presence of biofilms in paediatric medicine, specifically in acute recurrent tonsillitis. So you have possibly had your tonsils removed, maybe one of your children has their tonsils removed, and if you've went through that process, fairly unpleasant, but it maybe not quite as unpleasant as the disease itself. It's a very common disease, so it has a high incidence rate of 100 in 1,000 of the population, at least in the UK. And normally what happens is you present to your GP with a sore throat on multiple occasions, and if you're very fortunate, they might throw some antibiotics at you, but that's not necessarily the best course of action. And if you have more than 7 episodes of tonsillitis, at least again in the UK, you can be considered for tonsillectomy. The problem is that it's not as routine as an operation as you might consider, despite the fact that at least until COVID times, it has been fairly widespread. The current statistics are that more than 10% of patients that have their tonsils removed are readmitted for bleeding or post-operative bleeds within 2 weeks. And given that tonsillitis is normally considered to be a paediatric case and condition, this is affecting a young adult population who are an important career and social development point in their lives. They're missing school, and it can have long-term devastating complications. So ideally, we'd like to try and reduce the total number of tonsillectomies that are required. Now, the tonsils, in case you don't know where they are, those are the things that you're swabbing when you're taking a COVID test. So it's the palatine tonsils at the back of your throat. And rather than being the kind of smooth lumps that they appear to our naked eye, they have crypts that run deep in 3 dimensions throughout the whole organ. And we're working with clinicians in Glasgow who, based on early data published in 2003, and there's, I believe, something like an N of about 10 that suggests that there may be more bacteria present within cases of acute recurrent tonsillitis than previously thought. So this is one of these low N number studies where what you see in the high contrast image here is a biofilm located within a tonsillar crypt. So what we want to do is use the large field of view that the Mesolens affords us, the volumetric imaging, and try and understand, is this actually a physiologically meaningful result? Do we see more bacterial infection in these tonsils than we thought? So because we want to study tonsillitis, it means that we also need a control group. So our control group in this case is patients who have obstructive sleep apnea. So one of my PhD students, Megan Clapperton, shown here, gets the phone call in the morning saying that yes, there will be tonsils today. So she goes to the hospital, picks them up from our clinical collaborator, Katrina, drives them the 8 kilometres back. These are still living, we don't fix them. Performs gram-positive fluorescent staining using vancomycin Bodepi. And then we do dual wavelength mesoscopy using either reflection contrast to look at the tonsillar surface or the image using fluorescence contrast to look at the bacterial stain. So this is, again, it looks a bit like an N of 1 here, and I'll go on to explain a bit more about this in a moment. But if we press play in the movie, please. Thank you. The green contrast that you see here is this rather fluffy tonsil surface. And about halfway through the movie playing, there will be an overlay of the magenta false-coloured bacteria. And what you will see is they are almost everywhere. Now, that's a roughly 5 to 6 millimetre volume area that we're imaging. We are, in this data set at least, looking mainly at the tonsillar surface. But you can see that the bacterial infection and presence is indeed widespread. That's our N of 1. I'm just, for completeness, going to show you a completely separate data set taken from a separate patient on another day. If we can press play in the movie, please. Thank you. So again, we're looking at reflection contrast in green. Again, about halfway through, we're going to overlay some magenta stained bacteria. So you'll see in the tonsil surface, there are lots of bacteria. And as we zoom in, what we seem to see is a bacterial flock. We're possibly looking at a biofilm that's lodged within a crypt. Now, the prospect of us being able to see that with the light microscope is very low. But because we've got this large area of field of view, we don't need to scan around looking for one region of high bacterial burden. It's there in the data set. So at the moment, we're building up our statistics so that we can draw clinically relevant conclusions from this. We have not yet unblinded, so we don't know whether the data that I'm showing you today are from patients with obstructive sleep apnea or tonsillitis, but that's the next step. So once we've built up enough data sets to be able to do an unblind, we will do that. Then hopefully we'll publish it, so apologies to anybody, or thank you to anybody that gets this one to review. Last thing I'm going to speak about is a bit more technological development focused, at least to start with. And it's the problem of the amount of time that it takes us to image. So that whole lobe mouse lung that I showed you took about 7 days to image, because it takes us 5 minutes to image one image Z plane. That's with a pixel dwell time of 1/4 of a microsecond. And we can use that short pixel dwell time because the high NA, the Mesolens, gives us really high detection efficiency and optical throughput. Then for our Z series, we need to think about the number of Z planes and so on. So that's why it can take us a week to image one very large clarified volumetric specimen. So invariably we were presenting these data and people are sending us samples, and the throughput's agonisingly slow. And what we really wanted to do was to develop a light sheet illuminator. Except Gaussian optics is kind of not our friend. If we focus the spot size that's required for the thinness of the light sheet to match the depth of field of the Mesolens, unfortunately the beam just diverges either side and it becomes a bit too thick to use, or so we thought. Anyway, we thought we'd give it a go. So what we did was we built 2 light sheets, one of which is the Gaussian one that I've just told you about, and it turns out it's quite good for fast screening of large samples. And a second propagation invariant light sheet, which uses a Powell lens that's been placed in an off-axis position, shown by this red arrow here, and a combination of this offset in optical path with aspheric elements has produced a light sheet that's more than 4 millimeters long, that's around 8 microns thick consistently across that propagation length. So this is the work of Eliana Battistella and Jan Schneider, both of whom are looking for jobs in Boulogne and Freiburg, respectively. So if you're interested in hiring them, please do. You will not be disappointed. I'll now show you some data from Eliana's propagation invariant light sheet, where we've used this for a specific application in cell reactivity. So what you're looking at in this volume here is a mouse brain that's been isolated, fixed and cleared, but it's also been stained with an antibody against GFAP, which in our hands is a measure of cell reactivity and gliosis. So if we can press play in a movie, please. What you'll see is not very much. And the reason that you don't see very much here is because there's not a lot of cell reactivity. This is a naive specimen, it's a control, and there has been no infection. If we can perhaps advance to the next slide, please, and press play in the movie. Thank you. This is obviously a lot more fluorescent. Now, this is one of these trypanosome mouse models that I mentioned previously, where the mouse has been infected with T. brucei 21 days hence. And the fluorescence that we see, located mainly in the astrocytes, is an indication for us of inflammation within the brain. And because of the very high spatial resolution of the Mesolens, we can now, for example, make measurements of dendrite length, which has given us a sense of how much of these astrocytes changed in size in response to this challenge of inflammation. Now, key to the success of this light sheet is not just the illumination, but it's the amount of time that it takes us to acquire data. So rather than it now taking us 1 week to acquire data, these data sets were taken in only a few hours. So in terms of high content in combination with high throughput, we're now in a space where we can start to think about, for example, longer term, longitudinal studies, and in particular, thinking about drug screening. So with that, I think my time is up, and I would like to thank you for your attention. I'd like to thank everyone at Strathclyde for their help in supporting this work and their technological expertise, some of our UK and overseas collaborators. I've only mentioned some of them here whose work we've shown today. There are very many more. If anybody's interested in using the Mesolens, we'd be delighted to talk to you about how you might send us specimens and how we might work with you. And finally, I'll thank our funders, because without money, we can do absolutely nothing. So thank you very much for having me.